Welcome to the NanoCap podcast. I'm your host, Will Chernlin, and I'm excited to have you join me on this journey through the world of NanoCap companies. For the past decade, I've talked to thousands of business owners across various industries. It's been an enlightening experience revealing the vast tapestries of entrepreneurship and innovation that thrives beneath the surface of mainstream media coverage. Each week, I will interview different leaders of NanoCap publicly traded companies to learn about their history, what they do now, and their future. As a reminder, none of this information is financial advice, and it is all for entertainment purposes only. Let's start this week's episode. Hi, everyone. In this week's episode, I have the pleasure of speaking with David Platt, the CEO of Bioxytran, stock ticker BIXT. Bioxytran is a clinical stage pharmaceutical company that develops platform technologies in the field of glycovirology, hypoxia, and degenerative diseases to eliminate viruses and prolong lifespan using carbohydrate drug design. David, thanks for coming on the podcast.
Thank you for having me.
Before we get started, I would love to do a quick thanks to our friends Derek McCarthy over at INS Digital Intelligence who introduced us. David, do you mind telling me a bit about kind of your journey from the Weizmann Institute of Science to today? What were some stops along the way, and what kind of led you to be CEO today?
So after doing my postdoctorate, I came to University of Wayne State University and then University of Michigan, which I did research on complex carbohydrate chemistry and cancer and so on.
And then did you go into the medical field? Were you a researcher for a while, or what kind of led you into the business world?
In my background, I was connected with banking. I used to be in bank, work for a bank before I start my journey as a researcher. So I started my first public company in 1993.
Wow.
By getting into a public company, it was my first public company. At that time, it was rare since most companies in biotech were private. But I started as a public company, and then that's how I started, not through a venture capital, but through a public company.
And what was the advantage of you going left when everyone went right? Why did it make sense for you to go public opposed to stay private like everyone else back then?
It's a question of access to money. I thought it would be easier to access for research compared to venture capitals, which require more control, and I wanted to be in control in what I'm doing.
And what was that first company? What was it, a medical company too, or you said it was a biotech? What problems was it trying to fix?
It was the first biotech company out of 5. Now it's my 5th company, public company. Wow. So I started that one, and I took it from something like here, from OTC, few cents, like 20 cents, all the way to $33 and close to $1 billion in valuation.
Wow. Yeah, it's interesting because we've talked to on this podcast of a few different kind of biotech companies, and I'm always curious how they go from like a researcher to publicly traded company. But with you having a banking background, it makes a lot more sense that you had the banking and the researcher side. Why that was the easiest way for you to access capital.
Yes, and it works very well because I became a NASDAQ company, switched to NASDAQ, I think around 2 years later, I became a NASDAQ company. We were fully reported. It was another reason why I did it, because from the beginning, they accounted for every penny that you spend. It's all the disclosure. It's a great point when you raise money from investors, because everything that you do is get disclosed. And the last thing is not just the disclosure, is the ability to project and advertise properly what you do, since in the end of the day, this is a research company, it's not making money. So you depend on news. And by doing that through the proper channel, you advertise what you do, and it's easier to raise money.
No, that totally makes sense that you can kind of share your findings along the way, get people excited, and then have kind of new access to capital as things get better, opposed to having to raise a specific round and kind of keeping some stuff under wraps.
Yeah, but the problem is that you lose the support of venture capital. So you need to
Mm.
go to Wall Street type companies and not to raising money from venture capitalists. You will actually need to go to fund managers. Not always the stock price reflect. If your valuation is too low, not always they invest money. It's not everything is rosy. It's just another way to raise money. Maybe some people think it's more difficult because you need to have the reporting, it costs more money to report. But since I have experience in reporting and filing, so it was not foreign to me, I was very comfortable to become a public company. It's called early stage public company.
Makes sense. So let's fast forward kind of a few years to you accepting the role as CEO of Bioxytran. Can you give me a little insight into what made you want to be the CEO of this company and what Bioxytran does?
Because first of all, research over the years allow me to develop drugs. And the drugs are a major impact on human, number one. And number two, the interest of the continue developing a new technology, which nobody is doing, in a chemistry that nobody is doing. You are totally, it's breaking ground into your venture to making drugs for disease indication that with chemistry that nobody is really doing. In this case, it's a complex carbohydrate, which very few people are doing research and developing drug out of them.
And so what is it meant to hope kind of cure?
Over the years, what I developed, for example, the bioxytran depend on the name of bioxytran comes from biological oxygen transfer. So what I did, I developed a method, chemistry, when I take the hemoglobin, hemoglobin, as you know, it's from red blood, but carry oxygen. This is a molecule that exists in red blood cells. So I take the hemoglobin out from the red blood cells, and then there are about 300 million molecules of hemoglobin in one red blood cells. So I take the hemoglobin out and then take from the hemoglobin has two parts, has the heme, which carry the oxygen. It's a molecule by itself, and the globin, which is a protein chain that support the heme. So I take the heme out of the globin and I exchange the support of the globin with the polysaccharide. Now I have heme polysaccharide. So you say, why? Why? The reason is that for 100 years, people try to stabilize the hemoglobin to make it artificial blood or a carrier or molecule to carry oxygen. By doing that stabilization with the sugar, I actually created a stable molecule that can carry oxygen. And now when I use it in, let's say in animals, I can replace their blood. So it became a blood substitute or a way to oxygenate tissue. Now blood, right now, it's the only molecule or blood is only get stable to carry oxygen. You can keep it refrigerated for 21 days. That's it. You need to throw it away, and you need to have continuously fresh blood all the time. Here, by doing that exchange, what I described, taking the hemoglobin, take out the globin and replace it with polysaccharide, call it sugar or carbohydrate in, I create a stable molecule that can be stable for years. It doesn't need refrigeration. It's good for all blood types. There's nothing to do with there's no dependency on what blood type. So you can actually have, and it's very inexpensive because my source for the hemoglobin, for the blood, is from cow. So I have unlimited amount of material. As a consequence, I have a blood substitute, room temperature, good for all blood types, that can be stable for many years. And what is the come to do? I can treat ischemia or lack of oxygen, trauma, wound healing, stroke, things like that. There's a shortage of blood today around the world. Every country doesn't have enough blood.
So pardon my ignorance. So humans can use cow blood?
It's not cow. It's the heme is
Oh, the
similar
heme
in
is.
all
Oh,
heme. The globe heme is similar in all living creatures.
so it's just the one enzyme from the
The
cows.
heme is the heme
Yeah.
molecule is the piece that carry the oxygen and release that. So I take that and I put it on a polysaccharide. That's why I just use the cow because it's ready available and there's a huge amount of material that get
Oh, yeah.
wasted. So I have a raw material that I can take and make it artificial blood.
No, okay, that makes much more sense. It reminds me of like how they make insulin out of some pigs sometimes, type using their enzymes and such. So who is your main customer then? Would it be like hospitals and ERs, or do they use it more for like therapeutic things, or what's the best use cases so far you've found for it?
Hospital everywhere that you can put it in an ambulance. You have a ready available oxygenation material. The problem is that when people lose blood, there's no way to carry oxygen, and they die from lack of blood in case of trauma, for example. In the hospital, people getting blood all the time, so you don't need transfusion, for example. But there are many places in the world that you don't have access to blood. You die from lack of blood. There is no refrigeration, or it's a distance from the hospital.
Makes sense. Is it just by happenstance? Is there certain like countries in like Europe that are way better at like having blood on demand than other countries, or is it kind of just if you're close to a big city, you have a better chance?
All countries in the world, including US, has shortage of blood.
Wow.
Because we're talking about between 30 to 40%.
Oh, wow. Okay, I had no idea. So, where in the approval process is your product? And can you explain a little bit how the approval process works for, say, something like a blood substitute opposed to maybe just like a normal pharmaceutical?
We are preclinical on that technology. We have in the company, in Bioxytran, we have 4 different technologies, but on a platform. In this case, this is a platform by itself because you can make out of it, you can make treatment for stroke or blood substitute or anemia or ischemia in general or wound healing. We are preclinical. We did all the testing in animals, and just lack of fund prevent us from going directly to human. We also have an FDA-approved device to measure the oxygenation of the tissue by measuring the amount of oxygen used by the mitochondria in the cell. So we have the ability to test it in human, which was approved by the FDA. So all we need to have the drug to inject the drug, and then with the device test it. The device is
so
510K, approved by the FDA.
and so yeah, how big is this device, and when would you see it in market?
You freeze on me.
Oh, sorry about that. So is your device in market right now?
No, the device is not on the market. It was approved, but we have the machine, the device, but we never commercialized that. It was done properly for the drug
Oh
as a
yeah.
way to, because the device doesn't, we don't need to go to disease indication. The moment the device is approved, it's like having a blood test for sugar. So you have a drug to lower sugar in the blood, you measure the amount of sugar in the blood, and you say, this drug reduce sugar in the blood, that's why it will help diabetic patients.
Mm-hmm. Makes sense. So looking ahead, what are your long-term goals for Bioxytran? Is it to have your product stocked in every single hospital possible? That's the end goal, or where do you see yourself in the next 5 to 10 years?
The end of the company exit in terms of technology is always a license to a large company because we are a startup company or a research company. But if you look on the bigger picture, is having the drug or the blood substitute in every ambulance or every hospital. This is a very big technology.
Oh, it seems huge. And like you said, with the lack of refrigeration, I just think of all of like Africa and the Middle East and parts of Asia where it's hot and it's rural. It's almost impossible for them to probably keep any sort of blood on
Yeah, and
demand at any
also
point.
we're talking about in the US in terms of price. Insurance company charge about $1,000 or more for blood, every unit of blood from patients
Wow.
or from the hospital. You need to realize that insurance company get charged thousands of dollars of every unit of blood. By the way, even you give donation, it costs the insurance company with the donation. They will cover about between $1,000. There was recent article, $800 to $3,000 for unit of blood.
That's crazy. And is a
So
unit
we're talking
like a
about
half liter, or how big is a unit?
a unit is about 330 milliliters. So I'm talking about that some people need 10 units of blood. So it's like $10,000 just for the blood. And by the way, we make it since the way we're doing the technology, we lower the price. It costs us, $50 to make a unit of blood like that, artificial blood. We compete
That
with price. We will lower the price for that, for the whole treatment.
I had no idea that blood was so expensive. It's like a very, very, very fine wine. I had no idea that it was that expensive
Yeah,
per
the people
milliliter.
give donation, but of course, you need to process the blood. You need to clean it. You need to refrigerate that. You need to store it in terms of bags. It need to shipping and handling. So that cost, and you need to clean it and sterilizing that. Make sure there is no disease, AIDS, contamination, and on and on. So the unit of blood can cost $1,000 or more.
Crazy. Yeah, I had no idea. Well, thank you for all the information.
This is just one technology in the company. If you want, I can continue with the second technology that we developed.
Yeah, sure. No, I was one of the videos I was watching. You were talking about how up to 1 in 5 people suffer from long COVID. That you have a technology to help with that too. Is that one of the ones you were referring to?
Yeah, we developed based on the corona, in the middle of the corona, we developed a drug that actually in 24 hours in 24 hours you reduce the symptoms of the COVID, and in a couple of days, in about 3 to 4 days, we reduce the viral load to zero by PCR, by polymerase chain reaction, or the regular, testing for COVID. So that drug, it's also good for getting the debris of the virus from the human. It's still there in long COVID. So we are able to get rid of it for a long period of time. So give the patient a couple of months, the drug will eliminate the long COVID.
And that's something that I hear a lot about long COVID. Is it so with your drug? Is it something you just take the second you have COVID until your symptoms go away, or how does it work?
The potential drug, since it's not approved yet, but it's in the middle of clinical trial by the FDA, the drug itself get rid of the long COVID because it get rid of what is left there. Some people get the immune system, get the virus out of the system completely, or at least reduce it to a manageable level. But in some other people, it stay in the lymph node and other places. And by giving that material to actually eliminate those leftover viruses. So I'm trying to explain in simple terms for layman person to understand what we are doing here. It's a complex science. I'm not getting into the detail, but I'm saying that by giving that, we have the potential to eliminate the phenomena of long COVID, which is mean that the virus is still there and it replicates and you get rid of the virus. The chemistry works in a way that on the virus, on the SARS-CoV-2, that's the virus, there are receptors which I was working on them. I called them galectin in 1992 in my first article. By the way, since then there are about more than 10,000 articles, scientific articles on galectin. Since
Oh wow.
since I cloned that receptor, we develop in the company an antagonist or a way. It's a polysaccharide that recognizes those receptors and can bind to them. So by taking it orally, the drug, it goes into the system, bind to the receptors on the virus, and eliminate the virus, kill the virus.
No, and when did you come up with this solution? When did you kind of realize that long COVID was going to become a thing?
This when long COVID became a phenomenon after COVID, because we developed that for COVID and to eliminate COVID in the first place. But then we
Oh,
find out that patients with long COVID, when we anecdotally gave them the material, the drug, the potential drug, we find they got rid of.
oh, that totally makes sense. And then, did you mention you had a third technology that you guys are working on?
Yes, we have also third technology, which is a polysaccharide that I work on that for many years. And I find always different drugs that can, different modification of different sugar that can block cancer metastases. And cancer and metastases is depend, among other things, on I didn't find it originally. Galectin, those receptors are part of the metastasis process of the spread of the tumor. So by binding to those galectins, again, I can. There are, by the way, different galectins receptors. Then there are 15 are known. So the moment the cancer becomes metastatic or it has the ability to evade the primary tumor, it also upregulated or get more galectin on the surface. So by targeting those galectin on the cancer cells in combination with other drug, you can eliminate cancer metastases. And we have results in human clinical trial to show that we can block metastases with antagonist against galectin on cancer cells. Remember, the antagonist or the drug is made of polysaccharide. It's non-toxic by nature, and it's very effective to block cancer metastases.
And so you take it once you've recognized there's cancer and to stop it from spreading, correct?
Yes, when you go with that clinical trial, you will go toward the end stage cancer patient that failed all treatment and they metastasize. So you treat them to save their life.
Amazing.
We publish that they're peer-reviewed, and we anecdotally, it's in clinical trial.
So yeah, you kind of beat me to my next question. I was gonna thank you for coming on the NanoCap podcast as we come to our 30-minute mark. And yeah, where can people find more information on what you're working on, all your different papers? What's the best way to learn more about your company?
You go to the webpage. It's Bioxytran Inc. They will find all the information.
Awesome. We will link to that in the description, link to that on the website and such. Thank you again, David, for coming on the podcast. I learned a ton. There's all sorts of cool stuff you're working on, from helping people with cancer to helping people with long COVID, helping people bleeding out. You guys seem like you're trying to solve all the world's problems at once. So thank you so much for everything you've done and coming on the podcast.
Thank you.
This podcast is for informational purposes only and does not provide financial, legal, or any other advice. The information is not investment advice or any offer to buy or sell any securities or to make any investment. The views expressed by the guests are their own, and any reference to third-party products, services, or information does not constitute an endorsement, therefore, by NanoCap Podcast or its affiliates. The NanoCap Podcast Express disclaims all liability for any individual's use of the information presented in